Flies are an important species in science because they are a model organism. This lab was done using two sets of flies, male red-eye with no wings, or apterous, and female sepia with wings. The parent flies were mated and produced the F1 progeny. The F1 progeny consisted of 26 flies, 9 male and 17 female. All the progeny were red-eye with wings. After mating the F1 progeny, the results of the F2 were a total of 161 flies, 76 male and 85 female. 3 different types of flies were shown: red-eye winged, in which there were 108 total, 53 male and 55 female. The second type was red-eye apterous, in which there were 11 total, 3 male and 8 female. And sepia winged, in which there were 42 total, 20 male and 22 female. The results of the F1 and F2 progeny reveal that red-eye is dominant over sepia and wings are dominant over no wings. Flies are among the most important species on Earth. They are considered a model organism. They have many characteristics that make them ideal for experiments ranging from genetics to biochemistry. Their small size, short life cycle, and ability to be controlled are among those many characteristics. The flies are analyzed through their generations. It begins with the parent flies. They are mated and produce progeny labeled F1. And if flies from the F1 progeny mate, they are considered the F2 progeny, and so forth. The generations are key to deducing what traits are passed down from parent to offspring. Flies differ from one another because of their genotypes and phenotypes. A genotype is the form that an allele takes in an organism. Genotypes come in 3 different forms based on the dominance or recessiveness of the allele. The dominant allele is always expressed, and the recessive allele is only expressed if both parents pass it down to their offspring. There are 3 types of possible genotypes: homozygous dominant, meaning both parents pass down a dominant allele to their offspring; homozygous recessive, meaning both parents pass down the recessive allele to their offspring; and lastly, heterozygous, meaning one parent gave a dominant allele and one parent gave a recessive allele. Punnett squares are used to predict the percentage of offspring with certain genotypes. Given each parent genotype, we cross them using a Punnett square and can predict what genotypes the offspring will have. A phenotype is the physical manifestation of the genotype. There are both autosomal linked traits and X-linked traits. An autosomal trait is when the allele is present on an autosomal chromosome, and an X-linked trait is when the allele is only present on the X chromosome. X-linked traits are a bit more complex than autosomal traits in the case of their probability of getting passed down. For example, males cannot pass down an X-linked trait to their male offspring. In this lab, real flies were mated to observe their progeny and determine what traits are dominant and what traits are recessive, and if they are X-linked or autosomal. After each mating, the progeny of each were observed and recorded to find this information. To begin the experiment, the media was made. 1 scoop of potato mix and 1 scoop of water were put into a large test tube. Then 5 yeast pellets were added as well. Next, 10 flies were given. For this experiment, 5 male flies and 5 females were given. The male flies were red-eyed and wingless, and the female flies were sepia with wings. The flies were observed under a dissection scope. Once observed, they were put in the same test tube as the media. To keep them in a large vial, a sponge cover was used to allow air in. After 7 days, larvae could be seen growing on the walls of the test tube, as well as grown flies jumping inside. In order to efficiently observe those flies, fly nap was used. Using a small brush, it was dipped into the fly nap and placed in the test tube gently while it was on its side and left to rest for 5 minutes. After the 5 minutes, the flies were asleep and ready to be extracted from the test tube. To extract the flies from the test tube, it was gently tapped on the outside to loosen the asleep flies from their places. A small paintbrush was used to guide the flies onto the index cards. Once the flies were extracted, each fly was sexed under the microscope. Male and female flies can be distinguished visually. Males have sex combs that are dark and on their legs, and females do not. That is the most effective way to sex the flies. Another way that isn't as effective is that males have very dark abdomens that are rounded, and females have pointed abdomens that are not as dark. Once sexed, their phenotypes were also recorded. The number of progeny was recorded along with the number of each sex. Once all the flies were sexed, another test tube with new media was made. Five male and 5 female flies were selected from the F1 progeny to be placed in the new test tube. Once the F1 flies were selected, the remaining flies were disposed of in an ethanol bath. This test tube was left for a 7-day period to allow the flies to mate and produce the F2 progeny. After the 7 days, fly nap was used once again to put to sleep the restless flies within the test tube. After 5 minutes and the flies were asleep, the test tube was gently tapped on the outside to loosen the flies. To get them out of the tube, a small paintbrush was used again to get them onto fresh index cards. Once the flies were extracted, each fly was sexed under the dissection scope. Once sexed, the number of flies were recorded as well as their phenotypes. These flies are a part of the F2 progeny and were the final progeny recorded for this lab. The results for the F1 progeny were a total of 26 flies, 9 male flies and 17 female flies. All the F1 progeny were wild type, meaning they had red eyes and were winged. The results for the F2 progeny were a total of 161 flies. 76 were male and 85 were female. There were 3 types of flies produced. Firstly, the red-eye with wings, containing 53 males and 55 females. Secondly, the red-eye and apterous, or no wings. 3 males and 8 females were counted. And thirdly, the sepia with wings. 20 were male and 22 were female. There could have been a 4th type, sepia apterous, but none were seen. The first cross between the parent flies, red-eye males with no wings, and sepia females with wings, produced 100% red-eye with winged progeny. This means that the red-eye and wing traits are dominant over sepia and no wings. And because 100% of the progeny were red-eye with wings, we know that the males were homozygous dominant for eye color and homozygous recessive for wings, and the females were homozygous recessive for eye color and homozygous dominant for wings. These Punnett squares show the 4 crosses between the male and female parents for each phenotype. The letter A represents eye color, and the letter B represents wings or no wings. As you can see, we now know that all the F1 progeny have the same genotype, that being heterozygous for the eye color and heterozygous for the wing type. Since all the F1 progeny has the same genotype, we know the parent genotypes for the cross to produce the F2 progeny. These Punnett squares show the cross between the F1 progeny. These crosses produced heterozygous and homozygous recessive genotypes. The homozygous recessive genotypes represent sepia eye color or no wings, and the heterozygous genotypes represent the red eye color with wings. The F1 Punnett squares were true to the real-world results. 100% of the progeny were red eye with wings. The F2 progeny were also close to the Punnett square results. About 57% of the F2 progeny should have been red eye with wings. The real-world results were 108 out of 161 flies, producing 67% red eye with winged flies. The sepia with wings should have been about 19% of the progeny. The cross produced 42 out of 161, being 26% of the progeny. The red-eye apterous should have been 19% of the progeny. The results were 11 out of 161, being only 6% of the progeny. And the sepia apterous should have been 6% of the progeny, but none were seen. These varying results could be due to some flies not being counted because they were stuck in the media, or even some flies being counted twice. Also, there were larvae in each test tube when extracting the flies that were not accounted for because they were not mature enough to sex. But despite the results, we were on track with the Punnett squares and their results.